Welcome back to the Deep Dive, where we unpack complex ideas into essential insights. Today we're plunging into, well, the molecular marvels of biological organic chemistry, specifically Chapter 42 from Clayden, Greeves, and Warren's Organic Chemistry, the second edition.
Yeah, it's always humbling, really, to see how the fundamental principles we study, in the lab, organic
Sure.
chemistry principles, they're not just mirrored, but often perfected in living systems. So our mission for this deep dive is basically to pull out the really important nuggets about how life runs on chemistry. We'll be focusing on the intricate mechanistic details, those clever reaction sequences, functional group transformations, the precise control of stereochemistry, molecular geometry, and even nature's own approach to retrosynthesis. Get ready for hopefully some real aha moments. Nature is the ultimate chemist, basically.
Absolutely. Think of this as your shortcut, maybe, to understanding this complex chemical symphony that's constantly playing out inside us, around us,
Huh.
all through the lens of a top organic chemistry textbook. So let's unpack this. Okay, let's start with the basics. The foundational divisions. What exactly is primary metabolism, and how is it different from secondary metabolism?
All right, so primary metabolism is the core chemistry. It's common to basically all living things. These are the molecules you find everywhere, from single cells right up to us. Think nucleic acids, proteins, sugars, lipids, and key intermediates, things like glucose, pyruvic acid, citric acid, acetyl coenzyme, acetyl-CoA, and ribose. They're absolutely central. Life's essential chemical blueprint, you could say.
Okay, the universal stuff.
Exactly. Then you have secondary metabolism. Now, this involves chemistry that's less fundamental for immediate survival. It's often restricted to smaller groups of organisms, sometimes even specific species. These molecules often give plants their unique properties or maybe act as specialized agents. Defense, signaling examples, alkaloids, terpenes from plants, or certain steroids in animals like us. They're kind of the unique chemical signatures.
That distinction really helps frame it. Okay, let's dive into the information system. Nucleic acids. Chemically speaking, what are they made of? How do they work?
Okay, nucleic acids, they carry the genetic information, right? They're polymers, built from nucleotides. Each nucleotide has three parts: a heterocyclic base, a sugar, and a phosphate group. Adenosine monophosphate, AMP, is a classic example. Mechanistically, the phosphates are absolutely key. They form these stable bridges, the backbone of DNA and RNA. But crucially, they can also be built into high-energy molecules like ATP, adenosine triphosphate. ATP is super reactive because those phosphate groups are excellent leaving groups. It's a bit like how we might use 2-silyl chloride, TSCl, in the lab to activate an alcohol, nature's energy currency.
Right, activating it for reactions.
Exactly. And the bases, there are 5 main ones involved: 2 purines, adenine and guanine, and 3 pyrimidines, uracil, thymine, cytosine. What's really interesting is how these bases can be modified in nature. Caffeine, for instance, that's actually just a methylated purine.
Ah, methylation. That brings us to nature's approach. We might use something harsh like methyl iodide in the lab, but nature has a more elegant solution, doesn't it?
It really does. Nature uses this incredibly sophisticated molecule, S-adenosylmethionine, or SAM. That's its go-to methylating agent. And the reaction itself is a beautiful example of an SN2 reaction, that clean displacement mechanism. The sulfur in SAM is a soft nucleophile, the triphosphate's a great leaving group. Substitution happens easily at that primary carbon, and enzymes play a huge role, orienting everything perfectly for specific N-methylation. It's amazing functional group control.
Yeah.
Then there's the double helix structure of DNA Watson and Crick's discovery. It relies on very specific hydrogen bonding between those base pairs.
Yeah.
Adenine with thymine, AT, uses 2 hydrogen bonds. Guanine with cytosine, GC, uses 3. This precise pairing is fundamental for reliable recognition, replication, life's information processing. Now, here's a key stereochemical difference. DNA is more stable than RNA. Why? Because its sugar, deoxyribose, lacks a hydroxyl group at the 2' position. RNA has that 2' hydroxyl. And having both the 2' and 3' hydroxyls on the same side of the ring, it allows for really rapid alkaline hydrolysis. It happens through intramolecular nucleophilic catalysis. The base pulls off the 2' proton, letting the oxygen attack the phosphate. This difference in stability and reactivity, it's actually exploited in medicine.
So understanding this chemistry has real-world applications then?
Absolutely. This deep knowledge has directly led to antiviral drugs. Think AZT for HIV or acyclovir for herpes. These drugs are often modified nucleosides. Maybe they lack the sugar ring or have altered stereochemistry. It just shows how subtle structural changes, rational drug design, can have a massive impact on biological activity, disrupting pathways. Even cyclic AMP, CMP, a vital biological messenger. That's another nucleotide derivative, made enzymatically by nucleophilic displacement.
Okay, from information storage to the cell's workhorses.
Yeah.
Proteins. How are these built and what sort of chemistry do they do?
Right, proteins, polymers of amino acids linked by amide bonds or peptide bonds, as they're usually called in biology. The sequence is dictated by DNA codons, and the ribosome machinery adds the amino acids one by one. All the standard proteinogenic amino acids share a basic structure, same core stereochemistry, just differing in that R group side chain.
Mm-hmm.
Take glutathione, it's just a tripeptide, but it's found everywhere. The universal thiol, its job, detoxifying dangerous oxidizing agents. It sacrifices itself, getting oxidized to a disulfide. That's a key functional group transformation. Thiol, SH to disulfide, RSSR. It also acts as a nucleophile to grab nasty electrophilic carcinogens, tagging them for excretion, a crucial protective role.
It's incredible that such small peptides can be so vital and even inspire medicines.
Totally. Look at angiotensin II. It's a peptide hormone, raises blood pressure. ACE inhibitors like lisinopril, they're basically dipeptide mimics, cleverly designed. Lisinopril binds the ACE enzyme just like the natural substrate, but because of a modified linkage, it can't be hydrolyzed, so it just sits there, inhibiting the enzyme. It's a beautiful example of chemists tricking an enzyme, saving lives through targeted mechanistic reasoning.
That's amazing.
And beyond function, proteins like collagen give structure. Its unique triple helix needs specific amino acids like proline and hydroxyproline. Now, that hydroxylation, adding the AOH group, that's a functional group transformation that happens after the protein is made, post-translational modification, and it requires vitamin C. That's why vitamin C deficiency leads to scurvy. The collagen isn't formed properly, things literally start falling apart.
And antibiotics. Another huge area exploiting differences in chemistry, right? Bacterial chemistry specifically.
Exactly. Bacteria often use unnatural diamino acids, like D-alanine, in their cell walls, sort of a defense mechanism. Penicillin works because it mimics the D-ala sequence that bacteria use to build those walls. The key is penicillin's strained beta-lactam ring. It gets attacked by a serine hydroxyl group in the active site of the bacterial enzyme, the transpeptidase. That nucleophilic attack irreversibly inhibits the enzyme, covalently bonds to it. No complete cell wall, the bacteria just burst. It's a fantastic example of targeting a specific mechanism, a reaction pathway to kill the bacteria.
Wow. Okay, let's shift to sugars. Often get a bad rap, but chemically, they're much more than just energy, aren't they? What makes them so complex?
Oh, absolutely. Sugars like glucose and ribose, they mostly exist as cyclic hemiacetyls. That cyclization creates a new stereocenter, the anomeric carbon. So you get alpha and beta anomers, different spatial arrangements there. Glucose likes a six-membered pyranose ring. Ribose often prefers a five-membered furanose. And this equilibrium means that anomeric hydroxyl group can point in different directions.
And that leads to the anomeric effect, guiding reactivity.
Yes, exactly. When sugars form acetyls, say methyl glucoside, it's under thermodynamic control. And surprisingly, the axial OM group is often preferred. That's the anomeric effect. It's a stereoelectronic thing, lone, pairs on the ring oxygen, interacting with the antibonding orbital of the CO bond. It stabilizes that axial conformation. Understanding this helps chemists predict and control functional group transformations involving sugars.
And nature uses this.
Oh, constantly, and with incredible stereochemical control. React glucose with benzaldehyde, you get a single stereoisomer, a specific transfused bicyclic acetyl, but use acetone. It prefers to make five-membered acetonides, especially from cis-1-viryl 2-diols, so it can actually lock glucose into its furanose form by making a stable double acetyl. And if nature wants to trap glucose in its open-chain form, it uses thiols to make stable dithioacetals, different functional group transformations for different needs.
Interesting. And glycosides.
Yeah.
Or are sugars linked to other things?
Right. Glycosides link sugars to alcohols, thiols, amines, usually at that anomeric position, often helps with solubility or transport. You see alpha and beta glycosides, depending on the stereochemistry. A really cool example is cinegrin from mustard. It's a thioglycoside. Damage to the plant, an enzyme called a glycosidase hydrolyzes it. That releases a sulfur compound, which then does this amazing Beckmann-like rearrangement, carbon migrating to nitrogen to form pungent isothiocyanates. Ali-isothiocyanate, that's the mustard kick. Sulforaphane from broccoli is similar, thought to have anti-cancer properties, another vital functional group transformation.
So it just shows how versatile carbohydrates really are.
Immensely versatile. Vitamin C, ascorbic acid. It's derived from glucose, acts as a key antioxidant. Sucrose, table sugar, is a disaccharide. Then you have sucralose, the artificial sweetener. It's just sucrose with some chlorine swapped in. Makes it 600 times sweeter and we don't metabolize it well. Cellulose, a glucose polymer, but the linkages make it form these tough linear fibers with lots of hydrogen bonding. We can't digest it. And then amino sugars like N-acetylglucosamine, N-acetylgalactosamine, they add even more diversity.
Right.
Keton in insect exoskeletons, glycoproteins for cell recognition, stereochemistry and functional groups driving all this amazing structural diversity.
Okay, let's move to lipids. Cell membrane builders. What's key about them?
Lipids, things like glycerol, esters, glycerol triolate from olive oil is a typical triglyceride. They're fundamental. Now, glycerol itself isn't chiral, but its two primary hydroxyl groups are enantiotopic mirror images in terms of their environment. So if you modify just one, like making glycerol 3-phosphate, suddenly the molecule is chiral. That highlights an interesting stereochemical point, how modification can introduce chirality. Fatty acids usually have these unbranched chains, even numbers of carbons. They can have double bonds, often cis or Z. Those cis double bonds introduce kinks, which is really important for how they pack, especially in membranes, affects fluidity. When lipids hit water, the polar ester heads, like the water, the nonpolar hydrocarbon tails, hate it. So they arrange themselves forming membranes, or the micelles we see with soap.
And soap chemistry itself, saponification, is a classic functional group transformation.
It is. Boiled triglycerides with alkali, like sodium hydroxide. You hydrolyze those esters, you get glycerol back, and the sodium salts of the fatty acids, that's soap. Those carboxylate salt head groups love water. The tails love grease. They form micelles, trapping the greasy dirt inside, letting you wash it away. Basic organic chemistry in action.
It's always fascinating how nature achieves complex chemistry under such mild conditions.
Mm.
No Bunsen burners or strong acids needed usually. How does it do it?
Yeah, nature's toolkit is incredible. Take NADH or NADPH. They're nucleotide derivatives, basically nature's version of sodium borohydride, NABH4, our lab-reducing agent. NAD plus is the oxidized form, accepts a hydride. NADH is the reduced form, carries that hydride ready to donate it. Powerful reducing agent.
So how does nature get around the problem of enantioselectivity and reductions? Reducing a ketone in the lab often gives both mirror images, right?
Exactly. A classic example is reducing pyruvic acid to lactic acid. In the lab with NABH4, you get a racemic mixture, 50-50 of both enantiomers. But the enzyme, liver alcohol dehydrogenase, uses NADH and produces only one enantiomer of lactic acid, pure. How? The enzyme binds both the pyruvate and the NADH in a very specific orientation. It forces the hydride addition to happen from only one enantiotopic face of that carbonyl group.
Wow.
Exquisite stereochemical control in that reaction pathway.
Amazing. What about making imidioamines, reductive amination?
Similar story. Lab reductive amination usually gives racemic amines. Nature uses pyridoxamine and pyridoxal phosphate, derived from vitamin B6, catalyzed by aminotransferases. It's enantioselective and, importantly, reversible. The mechanism involves forming an amine, then some clever proton shuffling turns it into an enamine intermediate. Nucleophilic attack follows, then hydrolysis gives a single enantiomer amino acid.
And nature manages enolate chemistry without needing super strong bases like LDA.
Right. It uses clever equivalents. Lysine enamines and coenzyme ethyl esters are key players here. In glycolysis, for instance, the aldolase enzyme uses a lysine side chain. It forms an amine with dihydroxyacetone phosphate, which tautomerizes to an enamine. That enamine then acts as the nucleophile in an aldol condensation with glyceraldehyde 3-phosphate. Forms fructose 1,4,6-bisphosphate, again with precise stereo control.
And AL esters like acetyl-CoA.
Yeah, acetyl-CoA is huge. Thiol esters are interesting. They're less conjugated than regular oxygen esters. This makes them easier to hydrolyze, but also more easily analyzed. They form that reactive enol form more readily. That higher reactivity and tendency to analyze is critical for things like making citric acid in the Krebs cycle. The enol of acetyl-CoA attacks oxaloacetate, an aldol-like reaction, catalyzed by citrate synthase using histidine residues for acid-base catalysis.
Okay. And phosphenolpyruvate, PE, sounds important.
Oh, PP is crucial. A high-energy enol phosphate. You might think it comes from just phosphorylating the enol of pyruvate, but usually nature makes it via a dehydration reaction catalyzed by enolase. It's sort of an E1CB-like mechanism, pull off a proton first, then eliminate the hydroxyl group, even though there's a phosphate nearby. A very specific reaction pathway. PAP then acts as a potent enol source for CC bond forming reactions, or it can transfer that phosphate to ADP to make ATP, really versatile functional group transformation and energy carrier. And you mentioned retrosynthesis earlier. The shikimic acid pathway is a fantastic example of nature doing just that for aromatic compounds. Plants need to make aromatic amino acids, right? This pathway starts with an aldol reaction between PEP and erythros 4-phosphate. Then there's cyclization and oxidation and E1CV elimination. All carefully orchestrated steps to build up that aromatic ring precursor. It's like nature working backward to figure out the simplest starting materials.
Which brings us nicely to natural products, these specialized, often complex molecules made by organisms. What's the key to their incredible diversity?
Natural products, yeah. They come from secondary metabolism. We usually classify them by how they're made biosynthetically. Alkaloids, polyketides, and terpenesteroids. They can be anything from the poison in hemlock conine, that's an alkaloid, to the flavor of beer, humulene is a terpene. Amazing range of structures and functions.
Okay, starting with alkaloids. Amines, right? How are they put together?
Alkaloids generally come from amino acids. For example, pyrrolidine alkaloids like hygran or tropinone. They originate from ornithine, an amino acid, and acetate, which comes in as acetyl-CoA. We figure these pathways out using things like isotopic labeling, feed the organism labeled precursors, see where the labels end up in the final molecule. It's like nature's own retrosynthetic analysis puzzle.
Right.
The pathway to hygrin. It involves decarboxylating ornithine using pyridoxal phosphate, then N-methylation using SAM, cyclization, and then a chalazen condensation. That's where two acetyl-CoA units effectively join up via acetoacetyl-CoA, which then attacks an aminium ion intermediate to build the ring system. Tropinone comes from hygrin via oxidation. And what's amazing is that Robinson's lab synthesis back in 1917 actually mimicked this biosynthetic strategy, under mild conditions too.
Incredible foresight.
Yeah. Another big group, benzyl isoquinoline alkaloids, like papaverine from poppies. They're built from two molecules of tyrosine. It involves hydroxylation, transamination to intermediates like DHPP and DOPA. DOPA decarboxylates to dopamine. Then dopamine reacts with the ketoacid derivative, forms an aminium ion, which triggers an intramolecular electrophilic aromatic substitution, a manic-like cyclization, to form the core isoquinoline ring. Then some methylation and oxidation finishes it off. Complex but elegant pathways.
And polyketides, fatty acids and related compounds.
Right, fatty acids are built up from acetyl-CoA. But additional 2 carbon units are added as malonyl-CoA. The pathway involves a condensation step, then a stereoselective ketone reduction using NADPH, then a dehydration, usually an E1CB, giving a transalkene, and finally reduction of that double bond, again with NADPH. This cycle repeats, adding 2 carbons at a time, and some unsaturated fatty acids like linoleic and linolenic acid. Our bodies can't make them, the ones with specific double bond patterns, so they're essential fatty acids in our diet. They're precursors for vital signaling molecules like arachidonic acid, which leads to prostaglandins, thromboxanes, involved in inflammation, blood clotting. Aromatic polyketides like resveratrol from Greeves skins, also built from acetyl-CoA and malonyl-CoA, but using different cyclization strategies, often aldol reactions, to form aromatic rings.
Okay, and terpenes, pine smell, camphor.
Terpenes, defined by having carbon atoms in multiples of 5, C5, C10, C15, they all come from mevalonic acid. Mevalonic acid itself comes from 3 acetyl-CoA units put together via Clayden and aldol reactions, then reduced by NADPH. Mevalonic acid then gets phosphorylated and decarboxylated to give 2 key C5 building blocks, isopentenyl pyrophosphate, IPP, and dimethylyl pyrophosphate, DMAPP. These are the fundamental terpene units.
Right.
The reaction joining these C5 units, say IPP and DMPP, to make general pyrophosphate C10, is stereospecific. Nature controls the geometry. Now, making cyclic monoterpenes like limone or pine from journal pyrophosphate is tricky. Journal pyrophosphate has a trans double bond, which isn't set up well for direct cyclization to a 6-membered ring. Nature's solution, it's really clever, it involves an allelic rearrangement. The pyrophosphate leaving group shifts from a primary to a tertiary position. This allows free rotation around what was the double bond, setting up the right geometry for cyclization. A fantastic chemical trick.
And finally, steroids, cholesterol, hormones.
Steroids are also derived from mevalonic acid through those same IPP and DMAPP intermediates. They share biosynthetic roots with terpenes. They just highlight the incredible structural complexity and diversity that nature can build, starting from that simple C5 building block, using enzyme-catalyzed functional group transformations and cyclizations.
Wow. We really covered a lot there. A whirlwind tour through Chapter 42 of Clayden, Greeves, and Warren.
Yeah.
Diving deep into the organic chemistry of life, it really hammers home that nature is the ultimate organic chemist, doesn't it?
It absolutely does. And what's so fascinating, I think, is that there's no real magic here. It's chemistry. Nature uses the same fundamental principles we learn: nucleophilic attack, leaving groups, acid-base catalysis, precise stereochemical control, clever functional group transformations. The difference is the scale, the efficiency, and that incredible selectivity achieved by enzymes, all under mild aqueous conditions. It's just elegant. And that sophistication is a constant source of inspiration for chemists, guiding how we think about mechanisms, reaction pathways, even our own retrosynthetic planning.
It definitely is. Which brings us to a final thought for you, our listener. As we continue to learn from nature's ingenuity, its perfected chemical strategies, what other complex biological processes do you think will unlock using the tools of organic chemistry? What new applications, maybe new medicines or materials, might emerge as we keep digging into nature's solutions? Something to ponder. Thank you for joining us on this deep dive. Until next time, keep exploring the chemistry that shapes our world.